Hello everyone, welcome to India's largest online learning platform, ITLS Academy. The topic of today's lecture is the adult stem cell. So first of all, we will see the introduction of adult stem cell. Adult stem cells, like all stem cells, share at least two characteristics. The first one is that they can make identical copies of themselves, that is, they can self-renew for a long period of time. This ability to proliferate is referred to as long-term self-renewal. The second characteristic of adult stem cells is that they can give rise to mature cell types that have characteristic morphologies and specialized functions. The intermediate cell is called a precursor or progenitor cell. Progenitor or precursor cells in fetal or adult tissues are partially differentiated cells that divide and give rise to differentiated cells. The next point about adult stem cells is adult stem cells are rare. That is, they are not very frequently found. They are found very rarely. Their primary functions are to maintain the steady state functioning of a cell called homeostasis and with limitations to replace cells that die because of injury or any disease. Unlike embryonic stem cells, which are defined by their origin means the inner cell mass of the blastocyst. They are derived from the inner cell mass of blastocyst. Remember, we are talking about embryonic stem cells that we have discussed in our previous lecture. But if we talk about adult stem cells, so these cells share no such definitive means of characterization. That is, they are not derived from the or they are not defined by their origin. In fact, no one knows the origin of adult stem cells in any mature tissues. No one knows how these stem cells arise, how the origin of these stem cells, origin of these adult stem cells has occurred. Here you can see this is a diagram of stem cell. We have taken here an example of hematopoietic stem cell. When this hematopoietic stem cell divides, it gives rise to a same stem cell, that is a cell that is same in shape, size and functions same as the parent cell, but it also gives rise to a specialized cell, or we can call it as differentiated cell. This differentiated or specialized cell may be neuron, maybe any muscle cell, maybe blood cell, maybe RBC, maybe WBC, it may be any kind of cell. These specialized cells acquire their own function, their own shape and size, and their own ability to grow and mature. This was a proper stem cell. But what if it is a progenitor cell? What is progenitor cell? Here we have taken an example of myeloid progenitor cell. When this progenitor cell divides, it gives rise to two cells. Although this stem cell was also giving rise to two kind of cells. But these two cells was different. One was a stem cell and another was a specialized cell. But in the case of progenitor cell, it gives rise to a specialized cell and a specialized cell. So we can say that progenitor cell on the division does not give rise to any cell that is same in shape and size and function. Both of these cells generated by the division of progenitor cells are specialized cells. Few points are given regarding the adult stem cells. The very first is definitions of adult stem cells vary in the scientific literature range from a simple description of the cells to a rigorous set of experimental criteria that must be met before characterizing a particular cell as an adult stem cell. In order to be classified as an adult stem cell, the cell should be capable of self-renewal for the lifetime of the organism. Next point is, ideally, adult stem cells should also be clonogenic. In other words, a single adult stem cell should be able to generate a line of genetically identical cells. You can see here in this diagram, this stem cell is giving rise to specialized cells, and these both cells are cloned to each other. So here it is known that these both cloned cells are genetically identical. These cells, we are talking about these specialized cells generated by the division of progenitor cells. These are same in shape and size. These are both are specialized cells, which then gives rise to all the appropriate differentiated cell types of the tissue in which it resides. An adult stem cell should also be able to give rise to fully differentiated cells that have mature phenotypes, are fully integrated into the tissue, and are capable of specialized functions that are appropriate for the tissue. Next point is, the majority of researchers who lay claim to having identified adult stem cells rely on two of these characteristics. These characteristics are appropriate cell morphology and the demonstration that the resulting differentiated cell types display surface markers that identify them as belonging to the tissue. At present, there is, however, a paucity of research with a few notable exceptions in which researchers were able to conduct studies of genetically identical stem cells. These genetically identical stem cells are also known as clone. So now we will see some evidences for the presence of adult stem cell. What are the evidences? How we can say that adult stem cells exist? So the very first point is adult stem cells have been identified in many animal and human tissues. In general, three methods are used to determine whether candidate adult stem cells give rise to specialized cells. Candidate adult stem cells can also be isolated and labeled and then transplanted back into the organism to determine what becomes of them. Next point in evidence is it is often difficult, if not impossible, to distinguish adult tissue specific stem cells from the progenitor cells. It is very difficult to distinguish that a particular cell is an adult stem cell or it is a progenitor cell, and which are found in fetal or adult tissues and are partially differentiated cells that divide and give rise to differentiated cells. Differentiated cells are type of cells that have their particular function assigned. If we talk about the progenitor cells, these particular progenitor cells, so these cells give rise to certain types of cells such as the blood cells known as T lymphocytes, B lymphocytes and natural killer cells, but are not thought to be capable of developing into all these cell types of a tissue and as such are not truly stem cells. Next point in evidence is there are reports of endothelial progenitor cells. Endothelial progenitor cells are a type of progenitor cells. So, there are reports of endothelial progenitor cells, skeletal muscle stem cells, epithelial precursors in the skin and digestive systems, as well as some reports of progenitors or stem cells in the pancreas and liver. A term here comes that is plasticity. We are now going to see adult stem cell plasticity. What is this plasticity? Mostly plasticity means to be ready to withstand any unfavorable circumstances, that having the ability to differentiate according to their environment. If environment is favorable, then to gain any shape and size and function like the favorable environment, and if there is environment is unfavorable, then they can survive to that particular environment too. So let us see what is adult stem cell plasticity. The very first point is the term plasticity as used in this report means that the stem cells from one adult tissue can generate the differentiated cell types of another tissue. At this time, there is no formally accepted name for this phenomena in the scientific literature. So here are approaches for demonstrating adult stem cell plasticity. We are going to see what are some approaches to demonstrate this plasticity. So our point is to be able to claim that adult stem cells demonstrate plasticity. It is first important to show that a cell population exists in the starting tissue that has the identifying features of stem cell. This starting tissue will have the identifying features. After that, then it is necessary to show that the adult stem cells give rise to cell types that normally occur in a different tissue. Simply proving the existence of an adult stem cell population in a differentiated tissue is a laborious process. It requires that the candidate stem cells are shown to be self-renewing and that they can give rise to the differentiated cell types that are characteristic of that tissue. In the experiments reported to date, adult stem cells may assume the characteristics of cells that have developed from the same primary germ layer or a different germ layer. If we take an example, so for example, many plasticity experiments involve stem cells derived from bone marrow, which is a mesodermal derivative. Next point is, alternatively, adult stem cells may differentiate into a tissue that during normal embryonic development would arise from a different germ layer. For example, bone marrow derived cells may differentiate into neural tissue, which is derived from embryonic ectoderm. Except this, the vast majority of experiments on plasticity have been conducted with adult stem cells derived either from the bone marrow or the brain. The bone marrow derived cells are sometimes sorted using a panel of surface markers into populations of hematopoietic stem cells or bone marrow stromal cells. Next point is to study plasticity in vivo, that is inside the body. The cells may be dissociated before injection into the circulatory system of the recipient animal or injected as neurosphere. Here in this diagram, you can see how these stem cells differentiated. Here, this is a diagram of brain. You can see here CNS stem cell, that is CNS, that is Central Nervous System stem cell. This CNS stem cell is arise from the brain. Here, this stem cell give rise to blood cells. Further, these blood cells also giving rise to fat cell. Here these are bone marrow stromal cells. These bone marrow stromal cells can give rise to cardiac muscles. Further, this can also differentiate into skeletal muscle cells, epithelial cells, and neurons. These all are differentiated stem cells anyhow. These can also give rise to glial cells. Bone marrow stromal cells can give rise to glial cells, neurons, epithelial cells, and skeletal muscle cells. Here you can see this is bone marrow. This is bone, and bone have bone marrow filled in it. This highlighted portion is bone marrow, and this is red bone marrow. This bone marrow can also generate liver. Liver is also a site of stem cell, as liver cells have the property to differentiate, and these cells can give rise to same cells. So in our body, liver is a part that can be regenerated. So this is all about the adult stem cells. Our overview of the stem cells we have seen in this particular lecture. Thank you guys for watching this session. Follow ITLS Academy on various social media platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. For latest videos and updates, subscribe ITLS Academy on YouTube and press the bell icon also. You can get it on Google Play Store also. So thank you very much guys for watching this session.